Welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. Conor Liston will present Probing and Rescuing Dysfunctional Brain Circuits in Depression. BBRF is the world's largest private funder of mental health research grants supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of over 190 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has such great impact is because we do not limit our focus to one illness or condition. Since 1987, the foundation has awarded $475 million to fund more than 6,800 research grants around the world and across a broad spectrum of brain conditions, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Conor Liston. Dr. Liston is professor of psychiatry and professor of neuroscience at Weill Cornell Medical College. He is a BBRF Scientific Council member and a BBRF 2013 Young Investigator grantee. Our webinar will begin with Dr. Liston's presentation, which will then be followed by Q&A. To submit your questions, please use the Questions tab on the control panel. Please feel free to submit questions at any time. Following the presentation, I'll ask as many questions as possible in the time allotted. And now, I'm pleased to introduce Dr. Liston. Conor, the floor is yours.
Fantastic, problem solved. And we have plenty of time because I have like 30, 40 minutes of slides at most. So let me jump in. So, as you just heard, I'm a neuroscientist and a psychiatrist, and much of the work in my group has been oriented around answering what we think is a really basic and important unanswered question in depression neuroscience, and that is, what are the mechanisms that control the switches in mood over time that are kind of the defining feature of depression? Depression is, by definition, this fundamentally episodic psychiatric syndrome, meaning it's defined by periods of low mood interposed between periods of wellness. And most work to date has tended to focus on comparing a group of depressed people with a group of never depressed, what we call healthy controls, and doing so at a single point in time. And we've learned a lot from that work. But if what you're concerned with is understanding, why a person gets depressed today as opposed to last week or next month, and when they get depressed, what determines how long they stay depressed? And then conversely, when you treat them and they get better, what are the mechanisms that initiate that recovery? And are there perhaps other mechanisms involved in sustaining that recovery over time? And so that's what we're really trying to understand. And the goal is, if we can advance our understanding of these questions, we can perhaps develop better ways of treating people and achieving more durable recoveries for our patients. And there's very little work that has attempted to track patients over time. But one example that I really love, it's what you see depicted on the screen here. It comes from Bob Post and his colleagues over 20 years ago. They just followed a group of depressed people with unipolar and bipolar depression, and just described what they saw. And here you see three examples, three individuals. And periods kind of below this axis represent depressed mood. Periods above the x-axis represent what we call hypomania or mania, elevated mood. And 0 represents normal mood, what we call euthymic mood. And you can see just by glancing at these three individuals that they look very different. So you don't need any fancy statistics or any particular expertise. You can see that this person has kind of a characteristic length to their depressive episodes. This person's episodes are much shorter, but they flip back and forth more frequently. And they have relatively fewer prolonged periods of wellness compared to this person or this person. And so what we've been doing in the lab is following individuals with depression and repeatedly getting fMRI brain scans from these individuals as they cycle in and out of depressed mood states, and then doing analogous studies in mice using what we call two-photon laser scanning microscopy. I'll get to that in a moment, but this is an imaging technique that allows us to look at how microcircuits in the brain change in response to chronic stress and in response to antidepressants. Undoubtedly, many mechanisms are involved in these switches, but we think one of them involves the remodeling of these structures we call postsynaptic dendritic spines. Spines, which you can see here, they're these kind of thorny protrusions from the branches of neurons or brain cells. You can think of each spine as a kind of structural marker of a synapse, of a connection between brain cells. And we know that chronic stress, which can trigger depressive episodes in vulnerable people, we know that chronic stress is associated with a loss of these spines and brain connections. Conversely, we know that drugs like ketamine, which act as antidepressants, do the opposite. They lead to the formation of new connections between brain cells. And so it's been tempting to speculate that the formation of new connections is required for initiating antidepressant behavioral effects. And so in this first series of studies I'm going to describe, we set out to test that. Are these new connections required for driving mood state switches or behavioral state switches analogous to depression in animals? And we found that they were, but not in the way we expected. And I'm going to try to unpack that for you over the next few minutes and explain what it means for treatment. So before I jump into showing you what we observed, I just want to orient all the listeners here to the methods we're using. So we're able to visualize the remodeling of these spines, these connections between brain cells, by implanting a microprism into the brain and then using two-photon imaging, this microscopy technique, to acquire these very high-resolution images of brain cells and their connections in the living brain of mice. And you can see by using vascular landmarks, so the blood vessels and the contours of the prism, kind of like a window, we can return again and again to the exact same position with micrometer precision, so millionths of a meter precision. And we can ask what impact stress and antidepressants have on the remodeling of these brain cell connections. And in this first series of experiments, we implant a prism, and then we do imaging before and after various chronic stressors, and we ask what impact that has on the remodeling of brain cell connections. And you can see that again, you don't need to be an expert, you can just appreciate it by eye. The arrows here denote eliminated spines, eliminated connections. They're spines that are present in the first image, like this one, that are absent in the second image. They disappeared, they were eliminated after chronic stress. Conversely, formed spines are spines that are present in the second image, but weren't present in the first image, like those denoted by these arrowheads. And what we found was that chronic stress, in this case, chronic stress hormones in the drinking water, but other stressors do the same thing. They cause a loss of dendritic spines, a loss of connections on these brain cells in the prefrontal cortex. And interestingly, not shown here, you can just take my word for it, the loss of those connections didn't occur randomly. It occurred in specific positions in the neuron, and they tend to cluster together in particular locations on the brain cell, suggesting that specific connections to this cell, specific projections from other brain regions, might be targeted for pruning, for elimination by chronic stress in kind of a guided way. And perhaps that antidepressants are doing the opposite. And when we treated these same animals with ketamine or with a kind of saline placebo control treatment, and then imaged them a third time 1 day later, and we chose that time point because anyone in the audience who has any experience with ketamine treatment knows that after a single day, many patients report feeling that their mood is much improved after a single treatment of ketamine. And we find that in these animals, there's a growth of synapses, a growth of spines of connections between brain cells that occurs after ketamine treatment, consistent with the idea that ketamine and other antidepressants might be acting to reverse the effects of stress. And to test that, we went in and we classified each connection that formed after ketamine treatment as a restored spine, a restored connection, if, like this blue one, it formed in a position where a spine used to exist and then disappeared. Or we call these de novo formed spines, de novo connections, are those that form in a position where we didn't observe a connection in either of the previous two images. And what we saw was that ketamine had a dramatic effect on restoring lost connections after chronic stress. Importantly, I'm going to return to this in a couple of minutes, and this is kind of clinically relevant for patients. What I've just described to you is kind of the glass half full view of what ketamine is doing to brain connections. It's restoring many connections that were lost after stress. Importantly, though, about half of the connections that formed after ketamine treatment were restored spines, restored connections. That means that about half were forming in new random positions. And doing some quick math, we can estimate that ketamine is partially rescuing the loss of connections that occurs after stress, but not fully rescuing it. And we think that might be relevant for patients' clinical experiences in a way that I'm going to describe in a moment. I just wanted to flag it for you here. Okay, shifting gears, I'm going to tell you a little bit about what we see in terms of circuit function in these animals undergoing antidepressant treatments. And to test this, we use a method very similar to the one I just described, 2-photon imaging of their brain circuits through a microprism. But instead of imaging the kind of structural markers of connections between brain cells, we're imaging their functional properties. And you can see that in this video, where the neurons have been genetically engineered to fluoresce, to glow when they become active. And so you get these beautiful images that look kind of like Van Gogh's Starry Night, where neurons are lighting up and then disappearing and lighting up again. And we can use those visual readouts as a marker of how active the brain cells are. And our prediction was that if many connections between brain cells are being lost after chronic stress and restored after ketamine treatment, that we might see what we call functional connectivity, the degree to which the activity in a pair of cells is correlated. We might see that change after chronic stress and go back to normal after ketamine. And that's indeed what we did observe. This is a little technical, but I'll just sort of depict it because you can kind of see it again by eye. These matrices here, these squares with warm and cool colors, warm colors denote pairs of cells that are strongly functionally connected, and cool colors denote pairs of cells that are not strongly functionally connected. And you can see just by glancing at these that after chronic stress in this animal, there's a loss of warm colors, there's a loss of functional coupling. And when we treat the animals with ketamine, that those warm colors come back essentially. And the reason that we see these changes has to do with these plots here, which depict how active each cell is at a given time. And you can kind of see that a lot of the activity in this image occurs when all the cells represented in rows become active at about the same time. And you can see that chronic stress disrupts the occurrence of these ensemble events. And when they do occur, they tend to involve fewer cells. And again, we see that ketamine rescues that effect. So we think these ensemble events, what I just described as kind of functional effect of lost connections and restored connections after antidepressant treatment, we think they're behaviorally relevant in all sorts of ways. That includes fear learning, cognitive flexibility, the perception of social hierarchies and kind of reinforcing social interaction behaviors. I'm not going to talk about those today in the interest of time. I want to save time for questions. Instead, I'm going to talk about just two, and that is either motivated escape behavior. So how do you respond when you're confronted with a threat? Or motivated reward-seeking behavior. So what do we mean by motivated reward-seeking behavior? So we can measure this in both humans and animals in a variety of ways. This is one way we do it. This is work led by Rob Fecho, who's now a postdoc at Princeton. Rob adapted a task that had been widely used in other animals for many years to test reward-seeking behavior and its sensitivity to stress. In this task, the animals learn to navigate left or right in a T-maze to acquire a big reward by climbing over a barrier, or to require a small reward without having to expend any physical effort, without having to climb over that barrier. And we can adjust the relative ratios of the large reward to the small reward and the size of the barrier to see how much work each individual is willing to expend to obtain an incrementally larger reward. And summarizing a lot of very complicated work in just a single slide very briefly, Rob discovered that this particular circuit in the brain, which projects from an area of the brain we call the anterior cingulate to another area of the brain that's involved in reward processing called the nucleus accumbens, we find that this circuit is important for encoding a reward predictive signal that scales with effort. So it's larger when the animal expends effort in red than when the animal does not expend much effort in blue. And it turns out that by silencing this circuit during specific times during the task, we were able to show that silencing these cells after the mouse has already made a decision to expend effort and is now kind of collecting its reward, silencing those cells decreases the likelihood that the animal will make a decision to expend effort to obtain rewards in the future. We think this is really relevant in depression because we know that that patients with depression often experience what we call anhedonia, a loss of interest or pleasure in things that they used to enjoy. But we also know that if you encourage the person to actually engage in the enjoyable activity, they often do enjoy it. But the problem often lies in willingness to expend efforts to work to obtain the reward. You can imagine if you're depressed, even getting out of bed, getting dressed, walking to your favorite restaurant to spend time with friends, these things might feel very effortful and you might just choose never to engage in them. And we think that this circuit is very important for those kinds of decisions. And it turns out that after chronic stress, the animals very consistently stop choosing high reward, high effort options in favor of a low reward, lower effort option. And the degree to which they make those choices is coupled with the degree to which activity in this circuit is disrupted. And we're currently looking at how antidepressants like ketamine influence the function of this circuit. But we think that's one way it might be behaviorally relevant. Okay, shifting gears before I turn to tell you a little bit about our work using fMRI brain scans in people, I want to conclude this part by telling you what we learned about the role of those new connections that form after ketamine treatment. So what we found was really interesting and not what we expected. We thought that the formation of new connections in these brain cells would be required for supporting ketamine's antidepressant effects. And to test that, we did two experiments. The first one you see depicted here. We imaged the mice before and after chronic stress, and then again in the hours to days up to a week after ketamine treatment. And we asked which comes first, changes in the connections, changes in their circuit function, or changes in their behavior. And what we found, contrary to our prediction, was that the changes in behavior occur very quickly, just 3 hours after treatment. We now know that they occur even faster than that. Whereas the changes in the formation of new connections don't occur until 12 hours after ketamine treatment, peaking about a day later. And that means that the formation of these new connections couldn't be required for initiating ketamine's antidepressant behavioral effects because the behavior comes first before the new connections form. Not shown here, I'll also just tell you that we found that the circuit function changes parallel the effects on behavior and precede the effects on new connections. So again, they couldn't be required for changing the circuit function. But another hint for a different possible role came from this graph, which told us that the restoration of these lost connections, and you'll recall that restored connections, we define them as connections that formed in a place where we previously observed a connection between brain cells, and then that connection disappeared after chronic stress. We found that restored connections were tightly correlated with the maintenance of ketamine's antidepressant-like behavioral effect when we tested it 2 days or 1 week later. So we hypothesized maybe it's the case that these new connections aren't required for initiating ketamine's antidepressant effects. Maybe instead they're required for sustaining them over time. And to test that, we teamed up with a group at the University of Tokyo led by Haruo Kasai and Haruhiko Bito. They developed a new tool, a new technology that we can use to optically delete newly formed connections in the brains of animals in a very selective and kind of harmless way, and ask what impact that has on behavior. And in this experiment, we treated animals with chronic stress. Some of their connections disappeared. We treated them again with ketamine. Some of them were restored. And then we used the probe developed by our colleagues in Japan to optically delete those new connections and ask what impact it had on behavior. And what we found was consistent with kind of our second revised hypothesis. Chronic stress increased immobility in this behavioral test that is often used for screening for antidepressants. Ketamine reversed that effect, but if you delete those new connections, the effect wears off within a day or two. And importantly, we also had a negative control experiment where we deleted connections 3 hours after treatment. You'll recall that 3 hours after treatment, the new connections had nothing to do with ketamine. There were no new connections due to ketamine 3 hours after treatment. And so here we're deleting a random set of new connections, not ketamine-related connections, and you see that has no impact on the behavior. So what we think this shows and why it matters for patients is that the formation of new connections in this brain circuit isn't required for initiating an antidepressant effect, but it is required for sustaining it over time. And that's important because we know that if you treat patients with depression with ketamine, many of them, even most of them, will report feeling a lot better the next day. But if you don't do anything else to help them and you just wait a week or two, almost all of those people will get depressed again. And that's why in clinical practice, ketamine is usually delivered multiple times. And there's a lot of interest in what we might do to pair ketamine with other treatments, perhaps it's psychotherapy, perhaps with other medications or brain stimulation that could prolong the benefits of ketamine. And we think that this study suggests that other interventions aimed at boosting the formation of new connections might be helpful there, might be helpful for extending the durability of ketamine's antidepressant effects. So I'm happy to talk about that further in the Q&A. But before I do, I want to shift gears and tell you a little bit about our work in human patients with depression. This is work led by Chuck Lynch and Emmanuel Elbau, two former postdocs in the lab who now are running their own research programs at Cornell. Chuck and Emmanuel did this really labor-intensive and truly kind of heroic study in close partnership with participants with depression who committed to being involved in this study. We're very grateful to them for making this work possible. And they tracked a small number of people, 40, 60, up to 120 times now over a period of 1 to 2 years as the patients cycle in and out of depression. And they get these brain scans over and over and over again, along with detailed clinical assessments that allow us to kind of map what is changing in the brain as a person's symptoms change. And what you can see here in this heat map are a bunch of symptoms in rows that are related to anhedonia. And the darker colors depict times over the course of a year and a half when this person was more anhedonic. The lighter colors or kind of pale pink or white colors denote times when this person didn't really have much anhedonia at all. And just what you can see by glancing at this is that there was a lot of fluctuations. Sometimes this person reported very severe anhedonia. Sometimes this person had essentially no anhedonia at all. And because we're doing this dense sampling of patients repeatedly over time, we realized that we'd also be able to accomplish another goal, which is to understand how the topography, kind of the boundaries of these network maps change over time. We've known for some time that the brain is organized into functional networks depicted by these colors. But more recently, scientists across the country have discovered that the boundaries of these networks are actually quite different in different individuals, kind of like a fingerprint or even like a face. And we know that because human faces have certain features in common, we all have two eyes and a nose and a mouth in roughly the same position. And that's why you can average together a bunch of faces and get something that looks readily recognizable as a human face. But this average face glosses over all the individual differences that are so important in how we use faces. And it turns out that brains are the same. You can see that everyone has this yellow network, which we call the frontoparietal control network. Everyone has this yellow network in about the same position. But the precise boundaries of this yellow network are dramatically different across these different individuals. In some people, it's very small. In some people, it's much larger. And you can imagine how using a treatment like TMS, which stimulates the brain non-invasively through the scalp using magnetic field pulses, a treatment like TMS that is used to target a particular area of the brain based on what we call scalp heuristics, just kind of positioning the coil over the right place on your scalp, that treatment would engage dramatically different networks in these different people based on the topography of their brains. And so we wanted to ask whether these networks are systematically different in the brains of depressed people compared to people who've never been depressed. And what we found was really striking, and that was that one network, and only one network, is dramatically expanded in the brains of people who suffer from depression. This network is called the frontostriatal salience network. The salience network is important in orienting attention to emotionally salient stimuli in your environment. So that might mean things that are rewarding. It might mean things that are threatening or anxiety provoking. It might just mean like something unexpected. But in any event, this network is important for detecting those salient stimuli in our environment and orienting our attention to it. And you can imagine how disruptions in this network could be relevant in depression. We're currently investigating that. But what we know is this network is dramatically expanded in depressed people. Again, you don't need fancy statistics. You can just see it. It's much larger in Black in this individual with depression compared to this never depressed healthy person. We see it very consistently across many people with depression. This is 49 people with depression and 24 healthy control individuals. It's highly replicable, and the effect size is enormous. It's a much larger effect than what I am accustomed to in studying people with depression using brain imaging. In fact, it's so large that we can actually diagnose an individual with depression knowing nothing else about them except their brain network topography, except based on this brain scan. This effect has also already been replicated in other labs, so we can really be confident that it's real, and it's a solid foundation upon which to build a whole variety of new studies, which we're currently undertaking. So let me tell you just a little bit about what we know about it so far, and then we'll kind of pause and take questions. So one hypothesis that we had about why this might be important, what it might do to contribute to depression, it's important in orienting attention to emotionally salient stimuli. Perhaps it's the case that this network expands as a person gets depressed and then contracts as a person gets well again. And so we knew we would be kind of uniquely well positioned to test that hypothesis because we had these densely sampled data sets from individuals dozens of times as they became depressed and not depressed and depressed again. And what the data showed us were again, just the opposite of what we predicted. The boundaries of this network, this is one individual. You see 4 representative study visits. In these first 2 visits, the person is depressed. In visits 3 and 4, or number 10 and 13 here, the person is not depressed. And you can see that the boundaries of this network are rock solid stable. They really do not change almost at all with just like very minor discrepancies. We see that in all the patients we scan. The network topography, the size of the network really does not change. We see that it's unrelated to how many depressive episodes the person has had in the past, whether it's just a few or many. We see that it's unrelated to how depressed they are currently at the moment. And so that led us to, again, rethink our hypothesis maybe it's the case that this network isn't expanding and contracting as a person gets depressed and not depressed, but maybe it's somehow conferring risk for depression. So we kind of brainstormed for a while and thought, how can we test this idea that this expansion is important as a risk factor for depression? We reasoned that if that were the case, we should probably find that the network is expanded in individuals who have never been depressed, but later go on to become depressed, compared to individuals who were never depressed and kind of stay not depressed throughout the duration of study. To test that idea, we turned to a really unique resource called the ABCD study. This is a study that's been going on for years now. Over 10,000 kids have been enrolled in the study and then followed from ages, as young as 7 up to their teenage years. I believe some of them are approaching young adulthood now. And among those 10,000 kids, we identified 60 who were never depressed at ages 10 and 12, and then went on to become depressed in their teenage years, and had brain scans at both of these time points that were suitable for doing this functional mapping. And our hypothesis was that in these kids who were never depressed and scanned at ages 10 and 12, that their brain scans at age 10 and 12 should show an expanded network among these kids who went on to later become depressed, compared to individuals who never became depressed. And that's indeed what we found. The Salience network in these individuals was expanded in people who went on to become depressed compared to those who did not. So we're currently investigating, happy to talk about ideas about what that might mean and where it comes from, but we're currently investigating how it might contribute to risk. Importantly, we also found that the expansion of this network occurred differently in different individuals. I think in the interest of time, I'm just going to kind of briefly touch on this, gloss over it, and we can talk about it if it's of interest. But essentially, we see that when this network expands, it encroaches on its neighbors, the territory of the neighboring networks in different ways. And you can cluster the people in the sample, and you can identify at least 3 subgroups of patients based on what neighboring networks are being encroached upon. And we think that the risk conferred for depression may be related either to the expansion of the Salience network, or to the encroachment on its neighbors, or to some combination of the two. And I'm happy to talk about that further. We're currently incorporating this information into tools that we've developed for subtyping patients with depression based on fMRI measures. We know that these fMRI-derived subtypes are associated with different kinds of responses to transcranial magnetic stimulation, or TMS, for depression, kind of hearkening back to what I mentioned earlier, that different patterns of functional connectivity in these brain networks might be expected to respond differently to brain stimulation at a given site. And that's indeed what we see, that one of these subtypes is highly responsive to TMS. Subtypes 2 and 4 are kind of not at all responsive, and subtype 3 shows an intermediate level of responsiveness. In work that was led by Katie Dunlap, my former postdoc, who's now running her own lab at the University of Toronto, we now know that you can use a similar approach to model individual differences in response to SSRIs. And we think we can hopefully combine these things into a pipeline that would be used to first identify people who are likely to respond to first-line medications like escitalopram, Lexapro, a commonly used drug for depression, and give them the medication that we think will work. Or if we think that they're not very likely to respond to this medication, we could perhaps consider prioritizing second-line treatments like TMS and using this subtyping approach to identify what is the optimal target for treatment. And we're currently testing this strategy to see exactly how well it works. Hopefully I can come back in a year or two and have answers for you all on that. In the last 2 minutes, I'm just going to tell you one more twist to this story, which makes it a little more complicated, but also even more interesting, we think. I showed you that this network doesn't expand and contract as a person gets depressed and not depressed, but it turns out that the strength of connections between nodes of the network, they do change. In particular, the strength of connections between this area of the brain called the anterior cingulate and the nucleus accumbens, they fluctuate pretty dramatically over time. And the kind of loss of connectivity that we see between these brain areas is associated with different kinds of symptoms. So one circuit connecting the anterior insula and the accumbens is associated with anxiety symptoms. And another circuit connecting the anterior cingulate and accumbens is associated with anhedonia symptoms. And really interestingly, not only does it correlate with how anhedonic you are today, but we can get a brain scan and we can actually predict how anhedonic you are going to be 1 week in the future, suggesting both that this is probably playing an important causal role in driving people's anhedonia, and also that it might be an important place that we could intervene, perhaps with TMS, perhaps with other treatments, to prevent anhedonia from occurring, or from before, to kind of prevent it from getting worse before it kind of takes hold. And I'll just remind you that one way that we think this circuit is important comes from our work in animals, which I showed you about 15 minutes ago, showing that this exact circuit in the mouse brain is important for encoding a reward predictive signal that integrates information about effort and is required for reinforcing future decisions to expend effort to obtain rewards. So one can imagine how disruptions in connectivity in this circuit might give rise to subsequent problems with anhedonia and motivation. And with that, I'll stop. I just want to highlight all the people who contributed to this work, which you see photographed here, and their names highlighted in red, including my collaborators at various institutions. And I also want to thank BBRF and Jeff for their support over the years and for making this work possible. And I'll stop with that.
Well, Conor, thank you for covering so much in a user-friendly way. And these are complicated issues, and certainly for the work that you're doing, which is so impactful. I want to start off with a question from the beginning of your presentation about when you spoke about the effect of ketamine on the making new connections, bringing back, restoring connections. What do we know about other antidepressants? Do they do the same thing?
That's a great question, Jeff. I'll kind of speculate a little bit. I'll tell you what we know for a fact first, and I'll also speculate a little bit. So we know that other antidepressants, some other antidepressants, do the same thing. For example, we have a new study coming out showing that TMS, modeled in mice and animals, also forms these new connections. It does so in a very specific subtype of cell, which is interesting. And we know that experimental antidepressants like psilocybin is like a drug compound that induces hallucinations. It's kind of the active ingredient in hallucinogenic mushrooms. And some of you may know there's a lot of interest in psilocybin and other psychedelic compounds. Preliminary clinical trial data looks very promising for inducing rapid antidepressant effects. There's, of course, some work to be done still, not widely available, not available for clinical use yet. But psilocybin also forms these new connections. We know that other drugs that are not hallucinogenic, from work led by David Olson and others, but that have been kind of chemically engineered to lose their hallucinogenic properties, but maybe retain some of their other beneficial properties, we know that they also induce the formation of those new connections. What we don't know exactly yet is whether slower acting drugs like fluoxetine, like Prozac, or escitalopram, Lexapro, for example, whether these kind of first-line treatments induce new connections in precisely the same way. I think the field thinks that they probably do, but they do so probably through different mechanisms and on a slower time course. But I think there's a lot of reason to think that these new connections are kind of a final common pathway that is important across a range of antidepressants for maintaining antidepressant effects.
All fascinating and such important information. I want to jump to what you described with the ABC study, looking at the 10 to 12-year-old kids and being able to look at their brains and actually after the fact predict based on those scans that they would be the people who become depressed. Could you speak a little bit more about that and what that may mean in the future for potential treatment to decrease the risk of a person developing depression?
Absolutely. I love that question too. We're thinking deeply about that. So just to remind listeners, what we found was that by going to the ABCD study, we were able to identify a group of kids who are scanned at ages 10 and 12 and were never depressed, but later went on to become depressed. And we found that in those kids, their salience network was expanded compared to kids who were never depressed and stayed well during their teenage years. So that's consistent with the idea that this network expansion is somehow conferring risk for depression. We're really interested in why that is. You could imagine that somehow the alterations in this network are changing the way a young person responds either to rewarding stimuli in their environment or to threatening stimuli in their environment, or to a combination of the two. And that somehow may predispose them to becoming depressed in response to stressors. That's a hypothesis. We don't know yet, but we're testing that. Another idea is that when that network expands, it encroaches on the territory of neighboring networks that are important in cognitive control. Cognitive control is a term that we use to describe a group of processes that are important for basically like regulating our cognition, but also our emotional state. So you can imagine, if something sad or threatening is experienced by a person, you can respond to that in a variety of different ways. And one of the ways you can control how you respond is through cognitive control. CBT, different kinds of therapy, cognitive behavioral therapy, use cognitive control to kind of reframe how people respond to threatening or depression evoking experiences in their life. And we think that another way that this network expansion might influence depression risk is by disrupting that process, by limiting a person's cognitive control of their emotions. And we're looking into which one of those is possible. I'll pause in a moment, but I just want to address your question about interventions, what this might mean for young people with an expanded salience network. So a lot more work is needed to be done before you'd imagine scanning bunches of young people and telling them definitively about what this means about their risk for depression. But we do think that this information could be useful for some people. I want to emphasize we do not know, and I think we don't believe that this is the case either. We'll see what the data shows later, but it's not the case that I'm telling you that if you have an expanded salience network, you're guaranteed to become depressed. That's not true. But kind of like having high blood pressure puts you at risk of having a heart attack later, but doesn't guarantee that you'll have one. Knowing that you have high blood pressure can be a useful piece of information that might cause you to change other aspects of your life that would reduce your heart attack risk. For example, eliminating cigarette smoking, cutting back on unhealthy foods, exercising more. And so we think it might be useful for at least some families who have young people with an expanded salience network to have access to that information and to think about low risk things they can do that might that reduce their risk for depression in other ways. I wouldn't imagine taking a medication or going into a brain stimulation study just based solely on that. But you can imagine being careful with how you handle stressors in your life, perhaps having a lower bar for getting access to psychotherapy that might boost your resilience. So we're excited to explore that further.
Excellent. We, in other fields of medicine, and you spoke about it for heart disease, we focus on prevention by sort of knowing things in advance and taking action. For instance, blood pressure. There may be ways for us to help with prevention down the road using this important information.
Exactly.
You had said that you spoke about how with the ketamine treatment, the benefits dissipate with time, and there may be other interventions to continue the therapeutic benefit. Could you speak a little bit more about the research on that?
Yeah. So one thing that we have found, and that I think research is coming out on this, and it's consistent with how clinical ketamine is delivered. Repeated dosing of ketamine can be really beneficial for boosting the formation of those new connections. It actually works through two processes. So if you get multiple doses of ketamine, typically in a clinic, this might be done like twice a week for a few weeks. We know that multiple doses of ketamine form more of those new connections. And we also know that a larger proportion of those new connections will kind of get integrated into the circuit and will persist for the lifetime of the organism. So repeated dosing of ketamine is one strategy. But you might imagine other strategies too. For example, exercise. Exercise is something that is known to boost the formation of new connections in the brain. And exercise can assist with learning and memory. You might imagine that pairing exercise, at least in some individuals, with ketamine treatments, not literally at the same time, but perhaps they get ketamine on Monday and they do a little exercise on Tuesday and Wednesday. That pairing ketamine and exercise could be useful. We have preliminary data in animals suggesting that might be the case. That requires further study. Pairing ketamine with psychotherapy is another, I'd say, like hot area of study. The formation of these new connections might render the brain kind of more plastic, more responsive to the learning that takes place during psychotherapy, learning kind of new skills for promoting stress resilience. And so that's another idea. And perhaps pairing ketamine with other treatments that also form connections could be useful, especially in individuals with the most severe kind of treatment-resistant cases. So, for example, you might imagine pairing ketamine with TMS and seeing if you can kind of synergize between these two treatments that operate through very different mechanisms, but that both converge on the formation of new connections. That putting them together might be beneficial for enhancing the effects of both of them. And so we're currently looking at that too.
The all very exciting approaches. What if, when we do this conversation in 2 years or 5 years or 10 years from now, where do you see us going? Where do you hope and where do you expect we'll be in terms of our knowledge about these issues?
Well, I could talk your ear off about that. I'm going to try not to do that. I'm going to try to just highlight 3 areas that I think are ripe for kind of being transformed in the not too distant future. So one of them is developing new treatment protocols that just take our existing treatments and match them up with individuals more efficiently. Anyone on this call who has struggled with depression or has a family member or loved one who has struggled with depression knows that it can be a really frustrating experience, that much of what we do in treating depression, not all of it, but much of it relies on trial and error to some extent. We need to try a medication, see if it works, and if it doesn't, we need to try something else. And many of our treatments, as you all know, often take weeks or months to achieve their full effect. And so if you need to do 2 or 3 trials, that might be the best part of a year before you are fully better, or potentially even longer. And so we think that one way we can really help people is by developing new protocols that rely on biomarkers that might be fMRI brain scans. It might be digital phenotyping down the road, so measures that you can acquire, through passive sensing tools like rings and smartwatches and smartphones. It might mean peripheral blood markers, but objective measures that we can use and kind of integrate that information to formulate predictions about how likely a person is to respond to the treatments that we already have, that we know that work: medication, psychotherapy, ketamine, TMS, ECT, etc. And hopefully getting people to the right treatment much faster. That's one. Number two, we're really excited about the implications of the neuroimaging work for developing new ways of delivering TMS. I think of TMS not as a treatment, but as a tool for delivering a treatment, and it can be delivered in many different ways. And I think it's not controversial to say that we don't know the optimal way to deliver TMS, but it's probably not going to be exactly the same for different individuals, that there may be subtypes of people with depression who need one kind of TMS, and another subtype who needs a different kind of TMS. And we think we can learn a lot from this work about how to individualize TMS protocols, really generate new TMS protocols that are more tailor-made to your brain. And then the third, just very briefly, we're really excited about the opportunity to develop fundamentally new treatments, not just treatment matching or tweaking TMS protocols, but developing fundamentally new medications based on these insights about the role of new connections in the brain and the cellular and molecular mechanisms that give rise to those new connections. Where we think there's a lot of low-hanging fruit there, and we're excited to test that. I hope in a year or two we might have some clinical trial results to report, early phase clinical trial results, but that might be encouraging about fundamentally new treatment mechanisms that could help people.
Well, all of those are extremely exciting and sound all doable. And I just want to say to you, Conor, thank you so much for the work that you've done, for the work you are continuing to do, and thank you for joining us today.
Thank you. It's been my pleasure, and I hope we can do it again soon.
Absolutely. Absolutely. I want to ask people who are listening to the webinar to consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This webinar has been recorded. If you missed any portion or would like to share it with a friend or a relative, please visit the events and webinar page on our website. And join us again next month when we present another webinar on May 12th at 2 p.m. Eastern Time. Thank you, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Thank you.